recording? Good. Okay. Now, G proteins, we've described them before. There are a very diverse family of proteins that have certain common features. In fact, there's two major categories. We'll look at those in a little bit. Okay, G proteins bind either GTP, guanosine triphosphate, or GDP. Okay, that's one thing. Okay? As a matter of fact, they have to have one or the other bound to it, because if we remove it entirely for long enough, the protein actually denatures. That guanine nucleotide is essential to stabilize the tertiary structure. So you gotta have one or the other. Now, the other thing about G proteins. The G proteins have a different tertiary structure. In fact, it's often quite radically different, depending on whether GTP is bound or GDP is bound. So you have two structural states, a GTP state and a GDP state. So that's our second point. So you have different tertiary structure depending on whether GTP is bound or GDP is bound. For instance, if you had GDP bound here, you'd have that. GTP bound, you've got a different structure. And these would be the G proteins. Third point. All G proteins are GTPases. They're enzymes that hydrolyze GTP into GDP. Now, they're not very good at it. They actually are fairly slow, but they can hydrolyze GTP into GDP. Okay, so if you have a G protein like this with GTP bound, over a period of time, it will hydrolyze that GTP into GDP, toss out the phosphate, and switch into that other structural state. Now, in most cases, the GTP-bound state is the active state of the G protein. It's not always, but most of the time. And most of the time, the GDP state is inactive. Here again, there are exceptions to this. So, in other words, if a G protein binds GTP and is active, within a certain amount of time, it takes about 10 minutes or so, it will hydrolyze that GTP and shut itself off. Now, the fourth thing about G proteins. A G protein is capable of doing an exchange. It can throw GDP off and replace it with a fresh GTP from the environment. There's always a little bit of both inside cellular fluids, a few millimolar concentration. So we can do an exchange here. Okay, so we can do this exchange. We can throw out the old GDP and replace it with a fresh GTP. Now, a little misconception here. Although a G protein can turn GTP into GDP, it can hydrolyze GTP, G proteins cannot take GDP and turn it into GTP. Does anybody know why? Why can you only go one direction on it? Yes, if you hydrolyze GTP, that's a very exergonic reaction. It's very similar to hydrolyzing ATP. So that releases energy. All you have to do is catalyze it. On the other hand, the jam of phosphate onto GDP, that's a condensation reaction. That takes a ton of energy. G proteins cannot do that. they have to do an exchange. Now I'll give you another analogy here. Let's suppose the holidays are coming up, all right? And I want to get my wife something like a nice, snuggly nightgown. How about that? Now, being a fairly nice snuggly nightgown, okay. All right. Okay, now being a fairly prudent person, not quite sure what size she wears, I'm going to err on the side of caution and perhaps get the smaller of the two possible sizes, right? After all, I don't want to spend the next month sleeping on the couch, okay? Now let's suppose I get this nightgown for her and I decide to go for the smaller of the two sizes. So she gets it and she takes it and she says, oh, it looks so cute and stuff, and puts it on and she can hardly fit into it, okay? All right, well, safe, right? So what's she going to do? She's going to take it back to the store, and she's going to exchange it for the next larger size. So she takes it to the store, gives it up to the clerk, and then what's not going to happen, the clerk is not going to go and pull out a pair of scissors and cut that nightgown and then sew on another piece of fabric to make it larger, because it takes too much time and energy. So instead, she takes the smaller nightgown, and then you go to the rack and exchange that for a larger nightgown. That's what G proteins do. They can do an exchange, but they cannot turn GDP into GTP. That's endergonic. It takes too much energy. Okay, so that's a kind of thing. So all G proteins are capable of doing these things. So G proteins can exist in an active and an inactive state depending on whether they have GTP or GDP bound. Okay, questions on that? These are basic properties of G proteins. Now what we're going to do is we're going to kind of break the G proteins into two major categories and briefly, and then we're going to look at the sort of activation and shutoff cycles of these two types of G proteins. Now you can put these G proteins, there are literally hundreds of different G proteins inside our cells, each playing various kinds of roles. Many of them are involved in signaling pathways, but many have other roles as well. The first ones we have, what we call the monomeric The first was what we call the monomeric or single subunit G proteins. And they're often put into what we call the RAS superfamily. RAS is a particular monomeric G protein, very important to certain signaling pathway that we'll cover later. And a superfamily of proteins is kind of like, and any of you folks have those big extended family reunions once every year or two, where you get people from all over the state or all over the country, and you see your fourth or fifth cousins that you see once a year and that kind of stuff. That whole extended family, you can consider that a superfamily. You got, aunts, uncles, cousins, second cousins, third cousins, whatever. And they're coming from all over the place. They do share certain features in common. They have some genetic similarity. Even if you don't get along very well, whatever the case is, there is some common feature. That's like a superfamily of proteins. A big, huge, diverse, extended family that have certain key features. Now these monomeric G proteins tend to be about 25,000 molecular weight. They're fairly small proteins. That's one kind. And they play many different roles. You find some of them in signaling pathways. You find others that regulate part of the cytoskeleton. They're made of what we call actin microfilaments. They give cells shape, they move things around inside cells, they cause a lot of cell movement. Certain monomeric G proteins play roles in that. Others play roles in moving things in and out of the nucleus. Nuclear cytoplasmic transport. Okay. That's our monomeric G protein. They have many, many diverse roles inside cells. Now there's a second big category of G proteins. These G proteins are used exclusively in signaling pathways. And they are different. They're what we call the trimeric, or sometimes hetero, that means different, heterotrimeric. And that's our second major category. They're made of three distinctly different protein subunits joined together. In other words, quaternary structure. There is the alpha subunit, the beta subunit, and the gamma subunit. The alpha subunit is the one that binds GTP or GDP. In fact, the alpha subunit is structurally quite similar to the monomeric G protein, so it clearly came from a common ancestor, evolved in effect through the course of development of life on this planet from a monomeric G protein ancestor. But now we got these two additional subunits attached to them. These guys are used exclusively in signaling pathways. Now, one thing about these trimeric G proteins, one reason why they're used in many different signaling pathways is this. It turns out there are several genes that each code for a slightly different version of each subunit. There's about 10 genes for the alpha subunit and about 6 or 8 genes for the beta subunit and maybe about 10 genes for the gamma subunit. So in other words, there's about 30 different genes, each coding for one subunit or another. And the neat thing is this. The neat thing about it is this. You can take essentially any combination of alpha subunit, any beta subunit, any gamma subunit. Put them together and you get a particular trimeric G protein. If you use a different alpha subunit and the same beta and gamma subunits, you get a different G protein. So in other words, with about 10 genes for each subunit, that gives you about 1,000 different potential combinations. In other words, 1,000 or so different G proteins. And you get that vast diversity just by using about 30 or so genes in your genome. And the neat thing is this. Each one of those G proteins is going to be able to bind to a specific receptor or set of receptors and is going to activate specific cellular responses. So you have a whole bunch of different types of receptors. You can have a whole bunch of different specific G proteins, each one recognizing one receptor or another, maybe a few receptors, and each one of them giving a different cellular response. So that allows you to have a huge diversity of different types of signaling pathways. Things that recognize different ligands, things that activate different cellular functions, whatever the case is. So that's one, that's one reason why we use these trimeric G proteins so frequently in signaling pathways. Okay, questions on that, folks?